Hello. Um, Today is our second laboratory class of this semester, and we're going to briefly talk about principles of light microscopy. A compound light microscope is a microscope that uses a system of lenses where a lens close to the sample, these lenses that you see here, collects light passing through that sample to magnify that sample, create an image of that sample that is magnified. And then a second lens, that would be the, ocular lens or eyepiece lens. That one, further magnifies that image created by the objective lens. So basically, the use of a compound objective ocular lens combination allows for a much higher magnification. These compounds microscopes usually feature exchangeable objective lenses like you see here, allowing you to choose different objective lenses that give you a different magnifying power. This picture here shows a typical compound microscope. So you're already familiar with this, um, you know, type of equipment, but you have discussed this in previous bio class. So let's revise, the parts and their function. We're going to start from the bottom of the microscope to the top. Basically, you have here the light source. We call also that the illuminator. This is the light source for a microscope. Then right here, you have what we call the iris diaphragm lever. The function of this feature here is to control the amount of light passing through, the condenser. That's the next structure on top of the diaphragm. And, at the end, it's controlling the amount of light that reaches the specimen. It is located just below the condenser, okay? And the condenser is used to collect that light and focus the light on the specimen. It is located right here, you see, under the stage. And it's usually, positioned very close to the stage in order to get the best performance. This here is what we call the stage. This is where you place the specimen. And that also includes, these clips, stage clips that are used to move the slide manually on the stage. And then also the stage needs to be moved up and down when we're focusing the samples. And for this purpose, we have right here the coarse knob. This coarse knob is usually, is used with the scanning small objective to perform the initial, movements of that stage to get the sample on focus. And then after you achieve that initial focus and you start changing to other objectives with different magnification, then now you can use this other knob that you see here on top of the coarse knob. This one is called the fine focus knob. And that one is used to do the fine adjustment of the focus of the sample and allows you to do small movements of that stage to get that sample back in focus after you move after you move to another objective. Right here we have what we call the nose piece, and the nose piece houses the objective lenses. The standard objectives that you find in this compound microscope include, maybe a 4x. This is what we call the scanning objective that is used at the very beginning to focus um, that sample. Then you have the low power objective that is usually 10x. Then you have a high power objective that is usually 40x. And then finally the 100x that we use a lot in microbiology, that is also, it's also called the oil immersion lens. We will talk about this particular lens later. Then the other important part of that microscope, you see here on the top, the ocular lens. If you have a binocular microscope like that one, you will have two of them, one for each eye. And again, the function of this ocular lens is to further magnify the image that has been created through the magnification that is first achieved by these objective lenses. The magnification of these ocular lenses is usually 10x. So when we calculate the total magnification that you can achieve with this microscope, what you do is simply you multiply the magnification of the ocular lens by the magnification of the particular objective lens that you're using. Let's say if you're using the 4x scanning objective lens, then 4x multiplied by 10, the magnification of the ocular lens, that gives you a total magnification of 40x. But if you're using the 10x, 10x multiplied by 10x is 100x total magnification. If you're using the 40x objective, 40 multiplied by 10x for the ocular lens is 400 total magnification. And then finally, if you're using the oil immersion lens that has 100x magnification, if you multiply that by 10x for the ocular lenses, you can get a total magnification of 1000x. Okay. Let's now go through some key concepts related to microscopy, and we are starting here with magnification that we just, talk about. Magnification in a compound microscope, the total magnification or apparent increased in size is determined by the multiplication of the magnification of the objective lens and the magnification of the ocular lens. Another important concept in microscopy is resolution, resolution or resolving power. And this is the ability to distinguish two objects that are very close to each other. I will show you later how we can calculate this resolving power, but keep in mind that usually the best resolution you can achieve with a compound light microscope is around 0.2 micrometers. That is basically the minimal distance at which two objects can be in order to still distinguish them as individual objects. If they get any closer, you're using this microscope, if they get any closer than 0.2 micrometers, then the compound light microscope will not be able to distinguish them as individual objects. And now, they may look as a single fuzzy object that cannot be resolved by this microscope. And in that case, when you have these objects that are way, too close at a distance that is lower than 0.2 micrometers, you may need to use a different equipment for that. Let's say, for example, we're talking about distances in the range of nanomolar, for example, then you may want to use an electron microscope that can give you a way better resolution. Usually the resolution power of an electron microscope is about 1,000 times better than the compound light microscope. Another important concept in microscopy is contrast. Living cells are usually colorless, they're transparent. And then when you place them on top of a clear glass slide, there is very little contrast there to be able to distinguish them very easily. How do we solve this problem? Well, we apply stains, dyes, and these stains, they may color the cells or they may color the background depending on the kind of stains that you're using. We're going to talk more about staining techniques next week. So just wait only a week to discuss that topic. We continue here with another two important concepts in microscopy, the field of view and the depth of field. So in both cases, every time that you increase the magnification, that you move from one objective to another one that has a higher magnification power, both the field of view and the depth of field will decrease. Basically, the field of view decreases because an increase in magnification means that you are zooming into the sample to look at smaller, area. The depth of field decreasing with magnification means that you won't be able to focus simultaneously the whole depth of the sample if you keep increasing that magnification. Let's now take a look at one objective lens in this light microscope, and I just want to discuss the information that you usually see on these lenses, these numbers that you're seeing there. You have a set there of 4 different numbers. Let me tell you, what each of them mean. Well, first you have these on the top. The 10 right here means the magnification power of that lens. That means that this particular objective magnifies the object 10 times. Then if you look at this number right here, this is the numerical aperture. The higher that numerical aperture, the better the resolving power of that objective lens. What is the numerical aperture? Well, the numerical aperture is basically the ability of that lens to gather light and to resolve fine specimen details. The other two numbers that you see in the objective, this 160 is what we call the tube length in millimeters, and the other one, the 0.17. This is this refers to the thickness of the glass cover slip in millimeters, assumed by the lens designer for the best performance of the objective. Okay, so this would be thickness of glass covered slip, once again in millimeters. Okay, so I mentioned early on during the presentation, I mentioned resolving power, and I told you we're going to get back to that concept. And basically, this slide is showing you how you can calculate that value, that 0.2 that we mentioned before, 0.2 micrometers for a compound microscope. Where is that number coming from? Well, it comes from this formula that you see right here. This is the minimum distance between objects that can be seen as distinct. So this is basically the resolving power, right, that we're calculating. It has a constant value multiplied by the wavelengths um, and of light that is being used, and is divided by the numerical aperture. So if you look at this formula right here, if you want to have this distance between objects to be as short, small as possible, in order to have a better resolving power, because you want this number here to be as low as possible, because this is giving you the distance at which two objects can be next to each other without losing resolution. So if you want that number to get lower and lower, you want this wavelength length to be as low as possible. Let's say, for example, blue light, but not red light, that has a higher lambda wavelength value. And then you want this numerical aperture that is in the denominator to be as high as possible in order to get this number, as low as possible. That is the science behind that resolving power. So I mentioned to you that in microbiology, we use a lot one particular objective lens that is called the oil immersion lens, that usually has a magnification power of 100x. Why do we call this lens like this? Well, the answer to that is that in order for that lens to perform optimally, you need to add oil between the specimen and the lens. You see this oil here that is kind of like a bridge that is connecting the sample that is on top of the glass slide with the lens. Why do we use oil? If you try to look at, at a sample with the oil immersion lens without oil, when the light passes through the glass slide and then now goes into the air to then later reach the lens, when the light passes through these two medium that have a different refractive index, refraction is going to happen, and some of that light that is passing through the specimen is going to be lost. That information is not going to be collected by the lens as a result of that refraction. How can we solve that problem? Well, we add oil. Why? Because this oil has a similar refractive index to the glass slide. And as a result, when the light passes from one medium to the other one, from the glass to the oil, the light is not going to refract that much. And then now the objective is going to collect more information, more light, right? In other words, because that oil is facilitating the collection of more light, of more information, what that oil is doing is increasing the numerical aperture. It's collecting more light, it's collecting more information in order to produce higher quality image. So this final slide summarizes some rules that you should always keep in mind when you are using the compound microscope. You can revise them on your own. What I want to show you now is a video that gives you some good advices on how to use the compound light microscope correctly. Let's take a look at this video. Okay. So let's fix this.
Okay, now that you've made your stains, we're going to look at them under the microscope. I am expecting that you're in microbiology and you have the prerequisites and you have used a microscope before. So I am not going to go over the nitty-gritty details of how to use a microscope. I am going to focus on using a microscope for microbiology. If it has been a while since you've used a microscope or you've never used one, you will need to pay close attention to the details and make sure that you know what you're doing when you come to class. I'm not going to be able to spend a lot of time with students showing them how to use a microscope. Oil immersion I will help you out with because it's something new to all of us. So make sure that you understand what you need to do with the microscope, how to use it before coming to class. You don't want to spend an hour trying to find a stain. We have limited time and we need to focus on finding these bacteria as quickly as we can. Not saying that you should be so fast that you don't do it right. The other thing is we're going to use oil immersion. And with oil immersion, students get very, very frustrated. And don't be alarmed if you get frustrated and can't do it the first time. You will be required to do it, so just don't say I can't do it and I'm looking at my partner's microscope. That's not going to happen. You will be required to use oil immersion. You must use oil immersion for these stains, and you must find the bacteria with the oil immersion lens before you can leave the classroom. Now, as we get started, let's think a little bit back about what we've done. So we've made some stains, and remember that I had you draw circles to where you're going to put your stain, where you're going to put your smear. That was to make sure that when you look under the microscope, you're looking in the right place. When you look at this, you may feel like there's nothing on the slide, looking at it with the naked eye. Remember, bacteria are so small, you may not see your smear with the naked eye. So drawing that circle was so that at least when you put it on the microscope, you have somewhere to shoot for. So when we put it on the microscope, you put it in that clip, you put it over the light source. So if it's not centered, you're going to move it over the light source. One of the other things that I noticed right away is the light is really bright. Under the low powers, I'm going to want to turn it down. So I'm going to use the diaphragm lever and I'm going to close it a little bit by moving it to the right to kind of cut down on the light. The other thing that you want to look at is right here is the condenser. Sometimes the condenser is way down here and you will have focusing issues if you don't have it in the right place. So you're going to want to put it to where it's pretty close to the slide, right below it. Okay? Now we are dealing with bacteria that are very, very, very small. So we do not need to worry about the 4x power. We're just going to skip that. We're always going to start with the 10. Okay? You will have to find the organism, find the smear with the 10 power before you go any further. Okay? Now, to see if your condenser is in the right place, I have a little trick. So what you do is you find your specimen by using the coarse adjustment knob with the 10 power. So I kind of found it there. Now, to see if your condenser is the right place, take your loop, hold your loop over the light source, looking through the microscope, try to focus on the loop. It'll be a little bit blurry. So what you can do is you move the condenser until your loop comes into focus. Your specimen will go out of focus. Once that's done, that's exactly the perfect place for your condenser. So if you do that at the beginning of a lab before you do any microscope work, you're going to have better focusing. It's a little trick that I learned. It takes just a minute. If you skip it, you might have issues with focusing. Okay, so now I'm going to refocus by using my fine adjustment knob on my specimen. So now I found it with the 10 power. Okay, now I'm going to go to the 40. Notice that I do not move the slide. You may go, oh, it's going to hit. It's not going to hit. Okay, if you're in focus, these are parfocal microscopes. It's not going to hit. So now that I'm on the 40, I am not going to use the coarse adjustment knob. I can only use the fine adjustment knob. So my kind of rule of thumb is, in most cases, if you focus away from yourself, it will come into focus. If it doesn't, try the other direction. If you feel like you're just focusing, take a step back. Go back to the 10, readjust. Some things that can happen is you may not have it centered, okay? Make sure that you're centered right in the middle of the specimen, okay? So now I'm going to go back to the 40. Oh, I see it. It's a little bit blurry, but I see it. So I'm going to focus. Okay, going away didn't work, so I'm going to come forward. So it just depends on the specimen. One of the things that can occur is when you're making those slides, it's going to be a little bit cumbersome and you have a lot of different slides that you're working with. What can happen is students get a little bit out of sorts, I guess is the word, and they put their slide on upside down, okay? You will have focusing issues on the 40 or the 100 if your slide is upside down. So if you can never focus on the 40, go back to the 10, lower the stage, take the slide off and see if it's upside down, okay? Now I'm focused on the 40. I can now go to the 100. Now I don't go directly to the 100. You must use oil immersion. It is not an option. You may say, well, I can see it under the 100, which may be true. You'll see something blurry. Two things will occur. Either it will be the wrong color or it will always be out of focus. You must use the oil immersion to decrease the refraction of light so that you get a clear, visible specimen and it is the right color. When we do the gram stain, you are distinguishing between pink and purple. It may be pink without the oil, purple with the oil. The correct answer is purple. So you have to use the oil immersion. To use the oil immersion, I don't want to get any oil on this lens. The only lens that can ever have oil on it is the 100. Let me repeat that. The only lens that can ever have oil on it is the 100. Getting oil on the 40 or the 10 is very, very bad. It will permanently damage the microscope. So I do not want to put the oil on yet, but I do not want to go to the 100. So what I'm going to do is see how I'm turning it to where I have the specimen between the 40 and the 100. Now I'm going to take oil. It may be in a bottle like this. It may be in a bottle like this. Doesn't matter. If I use this, I have like a little medicine dropper thing, and you'll let the oil drop off. Okay? If you use this, it's a dropper bottle, and you just squeeze a drop off. Doesn't matter which one you use. You put the smallest drop of oil you possibly can. Do not touch the slide with the actual bottle or the dropper. Okay? Only if you let the oil touch the slide, it'll pull it off the dropper. Now that I have the oil on there, now I want to take a step back. Do you notice I did not move the stage? I did not move the slide. If you are focused with the 40, it will work with the 100. Make sure that it is absolute focus and it is centered on the slide before you put the oil on. It's kind of like the point of no return. So then I clip the 100 into place. So now I see it. It's a little bit blurry. Find focus. So I find focus and it comes right in. Now once again, if you find that you're focusing the heck out of it, you have an issue. Your slide's upside down, your condenser's in the wrong place. It may be that your microscope is dirty. We deal with those kinds of issues. Now, if you're focusing the heck out of it, do not use the coarse adjustment to focus, okay? If you're having issues, what should you do? Last time I told you to go back a step. I told you to go from the 40 to the 10. You cannot go from the 100 to the 40 because you will get oil on the 40 lens. We cannot have oil on the 40 lens. If you get to this point and you cannot find it, you must go back to the 10 to focus. Now you're in a situation where you can find it under the 10, but you cannot go to the 40. So you have to go focus, go back to the 100. Do not go through the 40 and focus again. This will be a little bit harder. So remember, make sure your specimen is perfectly focused on the 40 before you use oil immersion. If this doesn't work, going back to the 10, you're going to have to start over, which means you put it on a low power, lower the stage, clean the oil off the slide by dabbing it with a chem wipe, and then you'll have to start over. You cannot ever go to the 40 if there is oil on your slide. So once again, focus, focus, focus on the 40 before you use the oil. So now you've done all the right things. You have it under the 100. It's good and focused. You can find it. It's really important that you don't give up on this, okay? You need to make sure that you can find the correct specimen. You're going to see a lot of stuff on there. You may see big blobs of stain. One of the rules to keep in mind is bacteria is very, very, very tiny. If you have a cell what looks like a cell that's taking over the whole field of view, you are not looking at bacteria. It's a blob of something. Small bacteria. So don't be drawing things that are big blobs. That's the wrong thing. That's what I'll be able to tell from your drawings. You must find the bacteria because everything you do after this will be the same in terms of staining and finding specimens. If you can't find them on the first day, work at finding them. Because the next day you're not going to be able to find them, the next day you're not going to be able to find them, and you're going to be frustrated through half of the semester. So do not skip this and say, oh, I can't find it under the oil, I'm not going to do it. You must do it, okay? So now I'm done, I found my specimens, I've done my drawing, I'm done with the slide, okay? So what you're going to do, so remember it was way up there on the 100. You're going to go to a lower power, lower the stage, remove your slide. Now, we need to get the oil off of the 100 lens. To get the oil off the 100 lens, you use lens paper. Don't use any other kind of paper. Don't use a paper towel. Don't use a chem wipe. It must be lens paper. And I do not want you to rub it off, and I want you to be very gentle, okay? So you just take the paper and you dab, and you go to a dry spot and you keep dabbing until no oil comes off. Do you see how I'm just gently dabbing? I'm not pushing really hard and I'm not rubbing. I don't know if you can notice, but there's where the oil started, and as I dabbed, it got less and less and less. So if you dab and nothing comes off, that means you're done. Don't use a liquid to clean it off or anything. Now you're done looking at your specimen. Make sure that you keep your slides for just a minute. Talk to your lab partners before you throw them out, because you don't want, if their stain didn't want to work, they can use yours, okay? So keep them for a minute, talk to your lab partners before you, especially if you have a really good one, you want to share it with the class. Okay, now that I'm done, I want to put my microscope away correctly. So now I'm going to take it, always turn it to where the lowest power is facing down. You're going to turn off your microscope, lower the stage completely. You're going to center the little arm, and you're done. So now you unplug it, wrap the cord around the back. Probably don't need to do for you. You're going to put the cover on it, and remember when you take it back to the microscope cabinet, you carry with both hands. One hand will be holding the arm, the other hand will be underneath the base. Keep in mind the base gets really hot because of the light. So when you put your hand under there, it's going to be hot, but you still have to hold the microscope, okay? When you put it in the cabinet, the lens side goes in first. The, whatever this thing is called, the cord will be facing you. Always put it back in the spot with the number that is the same number as the number on your microscope.